Good morning, good afternoon, or good evening, whenever it is that you are watching this video. So this video is a catch-up on 8.3, which is applications of recombinant DNA technology that we discussed during the Google Meet. So this video is not meant to be a replacement for the Google Meet. It's only meant to deliver the main points. And if possible, please join the Google Meet in the following weeks. Alright, so in this video, I will give a quick review of tools in recombinant DNA technology and gene cloning, and I will also explain what is cDNA and how we use cDNA in the production of insulin in the lab. I will also go through a little bit about other applications of recombinant DNA technology, but we'll leave that one for later. Okay, so let's do that review if you're ready. So when we talk about tools in recombinant DNA technology, we're referring to the 5 things we learned in 8.1. So, okay, tools in recombinant DNA technology. Alright, the first tool that we need is target DNA, also called target gene, gene of interest, whatever it is. Basically what it means is I want to use this gene in the future, so I want to take it out and put it into a host cell so I can use it in the lab. So that's what target DNA means. Second, you will need a vector. Okay, if you're taking out your target DNA, sometimes you would need to put it in something else, and that is called a vector. So vectors, you may have understood this already, but I'll explain it again. Vectors are different from chromosomal DNA. Okay, chromosomal DNA is DNA that is very, very important to the organism. Without the chromosomal DNA, the organism wouldn't live. But vectors are smaller pieces of DNA that are kind of just like extra bonus DNA, you know. So if chromosome DNA, for example, in a bacteria is the CPU of the computer, the hard drive in the computer, then the vectors such as plasmid, cosmid, and also YAC, those are extra pieces of information, and we can compare them to external hard disk or pen drive or SD card. So vektor tu adalah cebisan DNA yang lebih kecil yang kita boleh gunakan untuk pindah-pindah maklumat dari satu organisma ke organisma yang lain. Kita boleh ibaratkan dia dengan pen drive. Okay, so there's different types of vectors. The one that we focus a lot on is plasmid because it's the easiest and we understand it the best. Next, what we will need after we have the vector is we need to have restriction enzymes. Restriction enzymes, there's so many types of restriction enzymes. Their function is they look for specific base sequences and then they cut at those specific base sequences. Dia cari, dia potong. Dia cari, dia potong. Itulah tugas dia. After restriction enzymes, we have modifying enzymes. An example of modifying enzyme is DNA ligase, which is what you learn about in chapter 6. Another modifying enzyme that you may have learned about in your books or in your notes is Tac Polymerase, TAQ polymerase. So Tac Polymerase is used in PCR, but PCR is not in your syllabus this year. So if you want to know more about it, you can look up videos about it. I will not explain it much in our class. And last but not least, you will need a host cell to carry, amplify, and express your gene of interest. And then, after you know the tools in recombinant DNA technology, you learned about the steps in gene cloning. So we can clone genes by putting it into a host cell and let the host cell copy that gene. Cloning means to make identical copies. So the steps involved in gene cloning, as you learn in 8.2, is first you isolate your target DNA and your vector. Next, you would use the restriction enzymes to cleave or cut your target DNA and your vector. Next, you would insert your target DNA into your vector. After that, you will conduct a transformation process where you take the recombinant DNA. Recombinant DNA bermaksud vektor yang sudah ada target DNA yang kita sudah gabung. Okay, you would put that into your host cell. If your host cell is able to accept that recombinant DNA, dia dapat terima recombinant DNA yang baru itu nama dia sudah berjaya transformation. Dia sudah berubah, dia sudah transform. Basically, that's what it means. And then you let the bacteria grow and it will amplify itself. And last but not least, you will conduct a screening test. Okay, screen is you do one step where you find out which of your bacteria are successful. Okay, is it successfully transformed? Did it take the target DNA ataupun dia dapat plasmid yang kosong? Itu yang kita cari dalam screening. So if you have questions about either of these either of these subtopics, then feel free to ask. Jangan malu bertanya ya. Alright, so what we're going to focus on next is we're going to talk about cDNA and insulin. So the production of insulin is an example of the application of recombinant DNA technology. If you don't already know, insulin is a hormone that regulates the blood sugar content. So dia akan kawal kandungan gula dalam darah kita. Okay, so it regulates blood sugar content. It is a hormone that is produced in the pancreas. In people who are diabetic, orang yang ada kencing manis, whether it's because of their diet or because of genetics, they are not producing enough insulin to control the level of blood sugar in their blood. So what we can do is we produce the hormone insulin in the lab, we package that into medicine, and then we give that medicine to the diabetic person. Mungkin bagi dia makan kah, bagi dia injection kah, tapi bila dia ada cukup hormon tu, badan orang yang ada kencing manis tu dia dapat kawal serba sedikit kandungan gula dalam badan dia. Okay, so insulin we can produce it in the lab to give a supplementary medicine to those who are diabetic. Now we're going to talk about how we produce that insulin in the lab. Okay, so for us to produce it in the lab, we are going to make use of the steps in gene cloning. But one thing we need to focus on is how do we get the target DNA? Target DNA kita adalah gene for insulin. The target DNA is gene for insulin. The host cell that we want to use today is E. coli. Okay, so I'm going to draw an E. coli right here. So our E. coli, this one doesn't have plasmid yet. Okay, so if you've realized something you might have, is that this gene for insulin came from human. Human is a eukaryote. And this host cell, E. coli, is a bacteria. It is a prokaryote. And that poses a problem because, I think you remember this one. The gene for eukaryote, the DNA for eukaryotes contain intron and exon. So, kamu ingat kan kalau manusia punya DNA, kita mau kasih jadi mRNA, dia dari pre-mRNA, kita buat RNA splicing, baru dia jadi mature mRNA. Sebab dia ada intron dan exon. Tapi untuk prokaryote is direct, straightforward. Dia cuma ada exon. So, kalau kita bagi direct, if we directly give them the human DNA, they don't know that they have to cut. Prokaryote tu dia tidak tahu yang dia perlu potong. Dan potong di mana, buang di mana, dia tidak faham. Jadi apa yang kita buat sebagai saintis yang pandai adalah kita tolong E. coli itu dengan kita buang intron itu siap-siap. How are we going to cut the intron when we cannot see the DNA? What we're going to do is we're going to take a more roundabout approach. Kita ambil, kita gunakan cara yang pusing-pusing sikit, tapi berkesan. Jangan risau. Okay, so what we're going to do is we're going to take it from mRNA. mRNA already contains only exons. And we're going to change that into DNA form. But that DNA, we don't call it normal DNA, we call it complementary DNA ataupun cDNA. Complementary DNA. Right, so let us take a few steps to understand how we can make cDNA in the lab. First things first, we look for the cells that produce insulin. Insulin is produced in the pancreas. This is a pancreas. What we're going to do is we're going to look for a very specific group of cells. This group of cells is called islet of Langerhans. Dalam seluruh badan kita, hanya kumpulan sel ini sahaja yang akan menghasilkan insulin. Only this group of cells in our entire body will produce insulin hormone. Bila kita sudah jumpa sel-sel ini, kita extract mRNA daripada dia. We're going to extract the mRNA for insulin from this group of cells. And then we're going to put that mRNA into a little test tube. Now inside this test tube I actually have a single strand mRNA that goes from 5' to 3' like this. Okay? Now, we're going to go into the next step to turn this mRNA into DNA. We're going to add some DNA primers, which is some free DNA nucleotides. And then we're going to add this very special enzyme called reverse transcriptase. Macam pernah dengar transcript ni kan? Okay, so reverse transcriptase, it will do the opposite of transcription. Kita tahu sudah bahawa transcription adalah proses menghasilkan mRNA daripada DNA. If transcription is using DNA to make mRNA, reverse transcription terbalik. Reverse transcription akan terbalik. Reverse transcription will make DNA out of mRNA. So if we look at this single strand of mRNA again, ini dalam test tube ni. Dalam test tube, kita tambah reverse transcriptase enzyme. Dia akan synthesize complementary DNA strand from 5' to 3' end. So this blue one is now our cDNA. And this orange one is still mRNA. Okay? First step. Very good. Now, I want to make the second strand of DNA and I want to get rid of the mRNA. Saya mau buang itu mRNA. Apa yang saya buat adalah saya tambah satu enzyme. Enzyme ini dipanggil mRNA degrading enzyme. Okay, so what this mRNA degrading enzyme will do is it will degrade the mRNA. Dia akan menguraikan mRNA kita. So what you will have is you will be left with a single strand of cDNA. Now this is cDNA. Okay, so to make that double strand, I am going to add another enzyme called DNA polymerase. So you already hear about DNA polymerase before. What it does is it synthesizes the complementary strand of DNA from 5' to 3' end. Okay, so dia akan synthesize dari 5' to 3', and now this is called a double-stranded cDNA. And there you have it. So now you have DNA that codes for insulin. And this blue thing is your target DNA. Sekarang kita sudah siap step menghasilkan ini. Dan kita sudah isolate kita punya target DNA. Alright, so let's go through the rest of the steps in gene cloning just to give you a refresh on how it works. Okay, so now we go back into gene cloning. First step, isolating the target DNA and also the plasmid. Ini saya keluarkan ya. Saya keluarkan pergi sini. So now we have our cDNA for insulin. And then we isolate a plasmid. Kali ini kita guna plasmid juga. Plasmid daripada E. coli. Dia ada AMPR, dia ada lexet. And then the next step is to cleave or to cut by using restriction enzyme. The restriction enzyme that we are going to use today is BAMH1. Ataupun dulu kami budu-budu, kami suka panggil BAMHI. Okay, BAMH1. BAMH1 will also produce sticky end. It's going to look for the sequence GGA TCC 3'. GGA TCC, and it will cut between the two guanine nucleotides. So if I add the restriction enzyme to our target DNA, it will cut right here and right here, produces sticky ends, and it will cut the plasmid right here. So kamu nampak di sini dia akan potong ngam-ngam di leg Z gene, dan kalau berjaya, kalau insertion kita berjaya, cDNA atau target DNA kita akan menyebabkan leg Z gene itu tidak berfungsi. Okay, so I'm gonna draw a plasmid. Forgive me if it's very ugly. Okay, so this is your AMPR gene, this is your leg Z gene, and that cDNA would sit right here. Okay, so this is your target gene. Lepas kita insert, kita mau masukkan plasmid recombinant kita ke dalam host cell. Okay, kalau plasmid yang sudah ada target DNA di dalam, itu kita panggil recombinant plasmid. So that recombinant plasmid will go into the host cell. Maybe it will succeed, maybe it will not succeed. So transformation. For transformation, under normal conditions, not in the lab, bacteria can do its own transformation in nature by using pilus. In nature, bacteria uses pilus to take extra plasmids in the environment or from other bacteria to transform itself. But in the lab, we force the bacteria to take up. We treat the bacteria with CaCO3 and temperature shock. Okay, kita akan kacau bully itu bakteria sampai dia punya cell membrane jadi permeable. By doing these two, we make the cell membrane of bacteria more permeable. Maksudnya, cell membrane bacteria itu lebih senang terima plasmid. So, we treat it with CaCO3 and with temperature shock with hope that it will take up the plasmid and successfully undergo transformation. After that, we let it grow. Okay, kita biarkan dia membiak. Bila dia membiak itu, kalau dia ada recombinant plasmid itu, dia akan duplicate recombinant plasmid juga. Dan kalau recombinant plasmid itu berganda, target DNA kita pun berganda. Dia kena duplicate sekali. Okay, and then after that we do a screening process. Screening is selecting which bacteria successfully receive recombinant DNA. Okay, so we're going to do the screening. Ini kamu sudah tahu dari last week. Kita ambil dia punya tinggi dari conical flask. Okay, kita ambil sini. Kita biarkan dia membiak di medium yang ada nutrient, yang ada ampicillin, dan ada X-gal. Okay, and then from here there are 3 possible outcomes. If you're not sure how this works, please ask me again. Okay, there are 3 possible outcomes. The first outcome is that the bacteria don't survive at all. The second outcome is that it does survive, and then it digests X-gal. If it can digest X-gal is a carbohydrate similar to lactose. So if it can digest X-gal means its LAC-Z gene is intact, masih berfungsi. Dan kalau LAC-Z gene dia masih berfungsi, bermaksud dia tidak mengandungi target DNA. So ini pun tidak berguna. Okay? If it can digest, it's useless. What we want is the bacteria that does survive and it is not able to digest X-gal. Because if it's not able to digest X-gal, that means its LAC-Z gene is not intact, and that means it will have the target DNA. So ini yang kita mau. Proses screening adalah kita mencari bakteria yang ada ciri-ciri ini. So once we have this, we can take that useful, successful bacteria and then grow it in the lab a little bit more. After we grow it in the lab, we can harvest the hormone insulin from this batch of bacteria. So when we talk about producing human insulin in the lab, that is an example of application of recombinant DNA technology. Itu adalah aplikasi ataupun kegunaan recombinant DNA technology dalam kehidupan kita. Of course, we use recombinant DNA technology to benefit us. Kita gunakan teknologi ini untuk memberikan manfaat kepada diri kita dan untuk semua manusia lah harap-harapnya. Okay, so the applications is in medicine. Application can be in medicine. Actually, I'll write this up here. This is an example of application medicine in the field of medicine. We can also apply recombinant DNA technology in agriculture, dalam pertanian. So in agriculture, we can use recombinant DNA technology yang ada plasmid, yang ada bacteriophage, ada vector, ada target DNA. Kita edit tumbuhan-tumbuhan kita, will form transgenic crops. Okay, transgenic means it contains DNA from other organisms. So kalau kita ambil DNA daripada tumbuhan yang rintang serangga, resistant to pests, is that how you say it? Okay, if we take DNA from another plant that is resistant to insect, we put it into our tomato plant, for example. That tomato plant which contains DNA to help it grow better, but the DNA come from other organism, that is a transgenic crop. And we can use that to, make our farming more effective. We don't have to plant so much to get only half the yield. We can plant less but get more food and longer lasting food. So the crops that we use the transgenic crops that we use in agriculture is also the widely debated GMO, genetically modified organism yang dia punya genetik sudah diubah-ubah. So it can benefit us depending on how we use it. And then you can also use recombinant DNA technology for the environment. So we have the problem with oil is, okay, di laut, di pelantar-pelantar minyak tu kadang-kadang pipe dia bocor lah, ada isu lah, and then you will have oil spills. What we can do is in the lab we create bacteria that can digest this oil and turn it into harmless substances, and then kita letak lah dia di lautan, and then hopefully they will be able to digest that oil and turn it into something harmless. Or we can use it in producing transgenic mosquitoes. Okay, you can edit the genes in other animals as well, so maybe the mosquitoes, you can edit it so that it dies when it is a larva. If you heard about the Zika virus a few years ago, virus Zika itu, they were debating on whether or not they should edit the gene of mosquitoes so that mosquito don't die, the mosquito will die. If there is no mosquito, the Zika virus, which travels using mosquito, will not be spread. So that is one of the uses of using recombinant DNA technology, our application for the environment. Another thing that you might not see in your lecture notes now, but I know it's a very possible thing, is using recombinant DNA technology to make bacteria that can digest non-biodegradable waste. For example, they can digest plastic. So my cousin is doing her master's in UMP, University Malaysia Pahang, and her project was finding bacteria that can produce specific enzyme to digest a specific type of plastic. So the project is underway. They are trying to grow the bacteria, and then hopefully they are able to spread the bacteria, let it live in garbage disposal sites in the landfills. So biarkan bacteria yang boleh hadam plastik itu duduk di tempat pelupusan sampah, dan harap-harap moga-mogaan beberapa tahun dari sekarang bacteria itu akan uraikan plastik-plastik tu, and that will help us a lot in the environment. So all of this are great things, it's wonderful things, but it comes with ethical and moral issues. So things that have to do with science usually comes with ethical and moral issue. Dari segi moral, in terms of moral and religious beliefs, there's always the argument that we are human, we're not supposed to be playing with this. Kita ni manusia, apa kau pandai-pandai pergi edit DNA kan? So in terms of religious beliefs, there's always the argument that we are playing God. Kita ni tidak sedar diri, kita ni manusia, kita mau buat kerja Tuhan, and that is not proper, tidak patut. Nanti ada might be a negative effect about this that we are not thinking about, or we are overstepping our boundaries, kind of thing. Another issue with recombinant DNA technology is GMO. A lot of people are thinking about, is it really safe? Okay, so this is a widely debated topic, and I think it would help you to form your opinion if you know more about GMO and what genes are actually being inserted into the transgenic organisms. Okay, bab-bab GMO ni bukan orang saja-saja untuk hasilkan tumbuhan-tumbuhan yang mutant, tapi mungkin ada efek yang kita tidak faham. Jadi apa yang kamu boleh buat adalah kamu belajar lebih sedikit apa gene yang digunakan, apa organisme yang dihasilkan. Go and understand the process of producing GMO for you to have a good informed opinion. Alright, next, another issue when it comes to recombinant DNA technology is we don't know what is the effect on the environment. Memanglah kamu hasilkan tomato yang boleh tahan lama, boleh tahan musim sejuk, berapa sejuk pun dia masih tumbuh juga. Tapi apa efek dia? Kalau kita biarkan tomato tu membiak, adakah dia akan terlampau bersaing dengan tumbuhan-tumbuhan lain sampai tumbuhan lain tidak dapat survive? Tomato saja. Kamu keluar tengok dari tingkap, tomato saja kamu nampak. Ataupun kalau kita produce transgenic organism bacteria, kalau kita produce transgenic animals, what will be the effect on the environment? If we produce transgenic humans, what will be the effect? We don't fully know. We cannot fully understand the future. So why should we be taking those big risks? That is some of the ethical issue that comes with this. And last but not least is that it will only benefit rich societies or rich people. So you know that there is a wealth disparity in the world. Kamu tahu, bukan semua orang ada bilangan harta yang sama. Ada orang yang betul-betul susah, ada orang yang dia tidak payah kerja pun dia hidup dengan senang lenang. Jadi bila kita ada kebaikan, kita ada kemajuan teknologi, kita ada recombinant DNA teknologi, there is a very big risk, ada risiko yang sangat besar yang orang-orang kaya sahaja yang akan dapat kebaikan dia. Only the rich may benefit from this brand new technology. And that is a very real possibility. Kalau bukan golongan orang kaya, mungkin satu negara itu saja yang dapat kebaikan dia. Negara yang kaya-kaya saja, negara yang miskin-miskin, negara yang sedang berperang, diorang tidak akan rasa kebaikan recombinant DNA teknologi itu. Satu lagi, dia bergantung kepada syarikat. Okay, it depends on the company. If the company is producing a GMO, dia kasih trademark dia punya GMO, dia ada hak cipta terpelihara, syarikat dia sahaja yang boleh hasilkan GMO ini. Kamu mau beli benih tumbuhan GMO ni, mesti beli dengan saya. Kalau tidak, saya saman kamu. So that company will create a monopoly. Only that company will be rich. Only that company will be able to get the benefits of the GMO products. And this has a very real example where only one company controls everything. One company monopolizes GMO products. I'm not sure if I'm writing the name properly. It's Monte Santo Company. Oh, sorry, it's Monsanto. It's the Monsanto Company. You can go and read more about it if you want. Okay, so things like recombinant DNA technology are great and amazing, and it can let us produce so many medicines, help us get rid of diseases, help us get more food to feed everybody, but it comes with issues. Just because we can do it, should we do it? So that is something for you to think about. There is no 100% right or wrong answer. And a lot of times we also don't know what is right or wrong. So get used to discussing and thinking about ethics and moral. You are very smart students, and I'm sure you can do a lot of things in science. But every step of the way, think about it. I can do it, but should I do it? And that is the ethics and moral issues. Okay? So I'll just recap. In this video, I've talked about tools in recombinant DNA technology. I've reviewed the steps in gene cloning. I have explained how we produce cDNA out of mRNA, and then how we use that to produce bacteria that can produce insulin. And then we also talk about other applications of recombinant DNA technology, and I hope you can understand a lot from this video. And I wish you all the best. Stay safe and take care. Bye. Thank you for your time.